Dizziness is a fairly common symptom that many people suffer from. Dizziness could mean general lightheadedness, but specifically for this experiment, we were interested in the sensation of vertigo. Now, vertigo is the illusion that your surroundings are spinning. It's due to asymmetry of neural activity between the left and right vestibular nuclei. Now, the vestibular system is located inside the ear. It's composed of semicircular canals filled with fluid and lined with sensitive hair cells. Positional changes cause movement of the fluid, which is sensed by those hair cells, and the hair cells communicate that information to the vestibular system, which provides you with a sense of balance and information about where you are in space. Now, when that system is damaged, people can suffer from things like nausea, vertigo, or just have problems standing up or walking. Vertigo can also be artificially induced by being spun around or even having water squirted in your ear. Now, because the vestibular nuclei are also important to vision, we were interested in testing out the effects of artificially induced vertigo on reaction time prompted by a visual stimulus. Specifically, we asked if there was a positive correlation between dizziness and reaction time. So to conduct our experiment, we used 3 subjects, and we conducted a baseline EEG for each of them. Though our focus was on reaction time, because EEG does measure the brain's spontaneous electrical activity, we were curious to see what type of effects the sensation of vertigo would have on EEG as well. So to induce dizziness, we had 2 subjects spin on a rotating chair for periods of 15 and 30 seconds, while the 3rd subject remained seated. Immediately after being spun, the subjects engaged in 2 types of reaction tests. Subjects would be able to rest for 2 minutes in between, and we then recorded a follow-up EEG for all of them. Our hypothesis was that there would be a relationship between dizziness and reaction time. Specifically, we predicted that reaction time would increase with increased spinning times and consequently increased level of dizziness. After obtaining results, we used a 2-tailed t-test to analyze baseline and trial reaction times. Now, for the first reaction test, we took advantage of the clicker function in LabTutor. LabTutor is able to record precisely when a subject presses down on the button and when he or she releases it. Responses are graphed, and the time between pressing and releasing is able to be observed. When a subject presses down on the button, the line on the graph goes up, and when the subject releases the button, the line goes down. Time is plotted on the x-axis, and for our experiment, we were only concerned with the initial pressing down. As a stimulus, we had subjects focus on the time, which runs across the x-axis and to the left. So we instructed subjects to press the clicker as soon as they saw an even number of seconds appear on the screen, meaning when they saw 2 seconds, when they saw 4 seconds, when they saw 6, 8, 10, and so on. We allowed subjects to continue pressing the button for about 15 seconds following each spinning time. The second reaction test was a little bit more straightforward. We found it on the internet. It involved clicking the mouse in response to a color change on the screen. So as you can probably see, this is the screen right over here, and in the center there's a red button. So as soon as that red dot turns yellow, the subject would click the mouse button as soon as you can, and the computer will record the time in between the color change and the clicking of the mouse, which is your reaction time. So you can see from this picture that the average reaction time in general was about 200 to 400 milliseconds. So what were the results? Graph 1 on the lower left hand here shows the results for 3 subjects performing the first reaction test. The times for the first subject, Jason, which is shown in blue, increased in the 0, 15, and 30-second trials from 200 to 300 to 400 milliseconds. The time for subject 2, Caitlin, shown in red, generally remained about the same, near 320 milliseconds, and the times for the control subject, Vinny, actually decreased across 3 trials. It went from about 160 to 80 milliseconds. Graph 2 on the right shows the 3 subjects performing the second reaction test in which you click the mouse. Subject 1, Caitlin, this time showed in blue, showed a slight increase from 280 to 320 milliseconds, but then it decreased down to 300. Subject 2, Vinny, showed a very slight increase from trial 1 to trial 3, with his reaction times ranging from 240 to 260 milliseconds. The control, Jason, also went from about 280 to 320 and then back down to 280 milliseconds. After performing a t-test that compared the baseline results to the 15-second test and the baseline results to the 30-second test, we found that none of the p-values were less than the level of significance except for one. That was Jason's results from the first reaction test on LabTutor, which did show a relationship between spinning time and reaction time. Now remember, his baseline reactions were 280, while his times for the 15 and 30-second tests were 360 and 400 milliseconds respectively. So it appeared that there was a positive correlation between spinning time and reaction time for Jason on the first reaction test. Data for the other subjects in the other trials did not turn out to be significant for this experiment. However, we did make several interesting observations while conducting the experiment. In the first reaction test, because we instructed the subjects to press the clicker when they saw an even number of seconds appear on the screen, we realized that it became possible to anticipate when the next even number would appear, namely after every 2 seconds. And instead of responding to the visual cue on the screen, subjects began to anticipate the number and press the clicker every 2 seconds. Some of the evidence for this occurring was that a number of clicks happened before the time actually appeared on the screen as shown by LabTutor. So essentially the subjects were pressing the button before the stimulus occurred because they knew when it would happen. So to account for these errors, we ignored any negative times, that is, when the subject clicked the button before the visual stimulus appeared. If that can be seen as a type of learning, we also observed another type of learning as well. Notice that in trial 1, subject 3, Vinny's reaction times, shown in the green, actually decreased. It went from 160 to 100 to 80 milliseconds. Because Vinny was the control and he wasn't spinning, we attribute his decreasing time to improved accuracy after several repetitions. Now, part of his improvement could have been due to what we described before, namely anticipation, but he described having to get used to the task over a period of time and then the task becoming easier for him to execute. And this was generally true for all of the subjects. So after a few trials, all the subjects appeared to become better at the test and their reaction times were affected. Another interesting observation that we made was that even though we had spun the subjects on chairs for 15 and 30 seconds, the feeling of vertigo would last only for about 10 seconds after they stopped spinning. Because we recorded reaction times for 15 seconds after spinning, the extra 5 seconds may have contributed to insignificant data. So, what were our conclusions? In general, we observed a positive correlation for one subject in the first reaction test only. For that subject, it appeared that increased spinning time, 15 and 30 seconds, did contribute to an increase in reaction time. Other trials with other subjects did not reveal significant data. We attribute the lack of useful data to several aspects of the experiment mentioned before, including anticipating the stimulus, improved reaction times due to learning, and a short amount of time experiencing vertigo. So for future experiments, we're curious to see whether increasing spinning times has any noticeable effect on reaction times. We would also like to see other types of reaction tests, such as those that rely on physical as opposed to visual stimuli, or tests that measure higher level cognitive functions, such as problem solving or reading comprehension. In addition to testing more subjects, we can consider the effects of other variables such as age, weight, food, that was consumed before the reaction test, as well as levels of hydration.